The discovery of the structure of DNA was published in 1953 by Watson and Crick. It led to a huge scientific endeavour around the world to work out the implications of why DNA was arranged in that way. Just how does the DNA molecule lead to protein formation?
You can think of DNA as being like a digital message. In the real world, there are 20 naturally occurring amino acids that make proteins. So any code in the DNA must be able to lead to the identification and organisation of the 20 amino acids.
DNA includes 4 bases, A, T, G and C. If each base identified one amino acid, only 4 amino acids could be identified, and proteins could only be built from those 4 amino acids. So the code has to involve more than one base at a time. What happens if the code were to use 2 bases to identify one amino acid? For example, if AA meant one amino acid and AG meant another. If this were the case, there would be 4 to the power of 2 possible combination. There would therefore be 16 amino acids involved in proteins. Clearly, this is also insufficient.
But if groups of 3 bases together were to code for a single amino acid, the number of possible combinations would be 4 to the power of 3, which is 64. This means 64 amino acids could be identified, and this is clearly more than enough to code for the 20 amino acids that occur in nature.
All research since has shown that every life form on the planet uses the exact same coding system. All amino acids are coded for by one or more groups of 3 bases. Some amino acids have a very specific single code of 3 bases. Others are represented by a small range of triplet codes. This is why there are 64 codes for 20 amino acids. There are also triplet codes that identify when the reading process should stop and start. The set of codes for a whole protein is a gene.
DNA can be viewed as the code strip for the amino acids that make proteins. But how and where does the code get read? DNA is in the chromosomes in the cell nucleus. Letting DNA out of the nucleus for protein synthesis would be a big mistake. Instead, the genes are copied, and it's the copy that leaves the nucleus. That way, the original DNA is never lost.
The molecule that does the copying is called RNA. There's a difference between RNA and DNA in that RNA uses the bases A, G and C, but not T. Instead, it has a base U. So if you look at a nucleic acid sequence and see the letter U, you know it must be RNA. If it includes base T, then it must be DNA.
Copying the gene on the DNA is called transcription. Inside the nucleus, the gene that's going to be copied is identified, the DNA unzipped, and complementary RNA bases are attached. The bases are complementary, so DNA base A joins to RNA base U, DNA base T joins to RNA base A, DNA base G joins to RNA base C, and finally DNA base C joins to RNA base G. Once the gene's been copied, there is a string of RNA with the complementary bases to those found on the DNA gene. This RNA is called messenger RNA or mRNA. Unlike DNA, it can leave the nucleus.
Once the mRNA has left the nucleus, it's in the cytoplasm. Ribosomes are organelles in the cytoplasm that enable the next phase, translation. This is where the code on the mRNA is read and the specifically identified amino acids are joined together to make a protein. For this to happen, molecules called transfer RNA or tRNA are also required. These molecules each have a group of 3 bases at the bottom using A, U, G or C. For each tRNA, the group of 3 bases is complementary to a group of 3 bases forming the mRNA code.
At the other end of the tRNA molecule is the specific amino acid that binds to that particular tRNA molecule. For example, tRNA with the bases A, C, C at the bottom attaches to the amino acid tryptophan. tRNA with either GGU, GGA, GGC or GGG attaches to the amino acid proline.
The mRNA from the nucleus attaches to the ribosome. A molecule of tRNA with bases complementary to the mRNA attaches and its amino acid is joined by a peptide bond to the previous amino acid coded for by the mRNA. The mRNA feeds through the ribosome and the process repeated for the next amino acid identified by the mRNA. In this way, a chain of amino acids grows to form a polypeptide or protein.
For example, if the mRNA code is GGG, ACG, the amino acids coded for are glycine and threonine. The complementary codes on the tRNA anticodons are CCC and UGC. So glycine attaches to one end of the CCC tRNA and threonine to the other. So our little pieces of mRNA codes for a protein made of these two amino acids.
In practice, the code is much, much longer, and the proteins are far more complicated than just two amino acids. The different number and sequence of amino acids leads to differently shaped polypeptides with different properties.
There is an important consequence of the code. If even a single base is mistakenly copied or missed, the amino acid that's coded for is different, and this directly affects the protein that's produced. The mutation might have no effect. It might be positive and make a change that makes the organism more successful. On the other hand, the mutation could be harmful and lead to problems for the individual.
Inside every cell, the DNA is always the same. The genes that are needed are switched on as and when required. For example, in all cells, the gene for a cell membrane is on, but the gene for the enzyme salivary amylase is only switched on in the salivary gland. Genes code for specific proteins, which can be structural proteins or enzymes, and are only switched on when needed.